Hey guys, so welcome back for the second half of our non-coding RNA session. So now we'll be talking about long non-coding RNAs. In the last slide set, we talked about short or small non-coding RNAs, which are defined as 200 nucleotides or less. Long non-coding RNAs will be 200 or more, so we can see that they are kind of a variable length. So this specific slide set was originally created by Dr. Andrew Nelson, who is now faculty at Boyce Thompson Institute, which is affiliated with Cornell. And if you're looking at the Live for Plants application, he is one of the primary mentors for the summer program. So this is kind of related to the types of projects you may find yourself working on if you're interested in this program. So let's go ahead and think about some of the things that make an RNA an RNA. So we'll start at the beginning. So the word gene, as we use it to describe a unit of DNA, was coined in the early 1900s. And we can think about genes as, again, being a unit of heredity. So we're thinking Mendelian genetics, which we've just come out of that chapter. So this is pretty relevant. Think of a heritable trait. We can take this sort of probability-based math approach to heredity. And now that we've been able to connect this up with our cytogenetics and cell biology a bit more, we know that these genes correspond not just to these sort of arbitrary elements, but they correspond to a place on a chromosome. We can then dive in a little bit closer, and we can see that there will be DNA that is going to encompass this gene. Of course, this can include the Tatabox promoter, and then we'll have the actual coding sequence, which could be made up of different exons and introns that can be spliced together. From there, we get our RNA. So this again is our messenger RNA, poly A tail, that's what this AN is kind of indicating. And if this is a messenger RNA, then of course we get a protein and central dogma. Okay, and this is, yes, this is a U and an A. Dr. Nelson used to work at University of Arizona, so it seems like this slide set probably could be updated a bit. Okay, so let's think about the level of protein coding genes that make up the eukaryotic genome. So the actual number of protein coding genes are a very small portion of the actual number of nucleotides we have in the human genome. So we actually only have about 25,000 known protein coding genes in a genome that is 3.2 billion base pairs long. So that's a pretty small fraction. Of course, these genes are, they can have various lengths, but just in the grand scheme of things, these protein coding genes we've kind of highlighted in blue here, that would leave an awful lot of junk between the genes. So this would be 100,000 nucleotides between genes or more. And you could think there's probably not a great reason to have that much just junk DNA, because there's a metabolic cost. If you think about replicating all of this and keeping it silent, like there's a lot of ATP involved with that. So why do we have all of this? Okay, so it's actually, the real picture is pretty different than this. So it's really complex. So we have protein coding genes and then small RNAs, small non-coding RNAs, like we talked about in the previous video, these kind of outlined in purple. And then we can have other types of non-coding transcribed regions that we're looking at. And over the past decade or so, we're able to see a really quick increase in the level of technology that we have to sequence these. So you can think back to those chapter 8 videos that have to do with sequencing and getting genomic sequencing done all at once. The technology has just really increased in the past decade. So these advances have allowed us to really collect a lot of information. So this can be on our left axis, we can see the actual number of base pairs of sequencing data in terabases. So this is a lot of data. okay, by 2018, which is approaching about where we are now. Okay, and we can also see the number of experiments that people have performed using this type of technology has really increased. And just for reference, you probably don't care, but here we go. I actually graduated from graduate school in 2012, and we were just starting to use this type of technology, and you can see how this has just really shot up even since then. So our orange is the actual base pairs RNA-seq data, okay, or RNA transcriptome sequencing, which we'll talk about in a bit. And SRAs are going to be the accession numbers for each of these experiments. So kind of can, in a way, kind of think of an SRA as being an individual experiment. So these are going to be archived and placed in a database like NCBI that we've used when we looked up differences in COVID spike protein variants. So we've been able to see lots more information that's given us a picture that we probably have more of these transcribed regions that don't code for proteins than we thought, maybe even more than that, and potentially even more than that. And it turns out that most of these non-coding transcribed regions are what can be described as long non-coding RNAs. So I think maybe more than 85% of the real estate in the human genome is actually taken up by these mysterious long non-coding RNAs. So you probably are getting a good idea of what a long non-coding RNA is, but let's define it. Again, long non-coding RNAs are not going to code for a protein. So they are transcribed. They do have a gene present some way, shape, or form in the DNA, but they're going to not code for a protein. So they're greater than 200 nucleotides, which will distinguish them from our smaller non-coding RNA. And you can see they have a 5' cap and a poly A tail. On the right, we've got our protein coding gene. This is a regular messenger RNA. See in blue, 5' cap, poly A tail. These can be variable lengths, okay? So you can have some that are, small. You can have some that are longer. But they have definitive start and stop and coding potential. So like you might think that what's in here might actually make a protein. And we can use these factors to distinguish the protein coding genes from the long non-coding RNA genes. Okay, and you can see that we're going to have other factors, because what's to say if this doesn't make a protein, what's to say if this just isn't random? We're going to be trying to also distinguish whether these long non-coding RNAs are what we call transcriptional noise, or just kind of like transcribed almost for no reason. But usually we'll be able to pinpoint that they might have a purpose based on whether they are going to be expressed in a specific cell type or tissue. If they have different expression during different phases of development, or in response to environmental cues. So those are some of the big ones. We have a couple of examples coming up, but there are actually several more that I'm not going to cover in this particular slide set. And you can see the structure of these. We can also classify some of these by their genomic context. So hopefully you're kind of noticing that they're positioned oddly. Okay, we can have, of course, just over here by itself, we can have long intergenic non-coding RNA, so between genes, so just here by itself. Okay, we can have kind of over here that's sort of loosely associated with the gene, so we can, sometimes they call these enhancer RNAs, but it just depends on what it's doing. You can actually have them as antisense non-coding, which means we would read it from and transcribe it from the opposite strand. Remembering that DNA has 2 strands, we might consider the protein coding gene to get the sense strand in this direction, and then our actual long non-coding RNA would start somewhere within the tail end of the protein coding portion, and it would be transcribed on the opposite strand in the opposite direction. So we call that antisense. Kind of overlapping underneath. This would be like the upside down of our transcription. Or we can have them start within the intron of a protein coding gene. So we can categorize these in a lot of different ways. So a lot of the things that we thought previously were junk, not really junk. And the lncRNAs actually heavily outnumber protein coding genes, and the classifications, there's so many of these that really, and there's no coding information. Sometimes it's really hard to get a lot of information about what these could be doing in the genome. So again, these are sometimes difficult to distinguish between sort of junk transcription or transcriptional noise that has no biological function, because sometimes we don't see lncRNAs actually being conserved across species. So it's not like you have a protein like hexokinase. You can find that in like every organism. lncRNAs are kind of species or closely related species specific. They're not transcribed as heavily as many of the other types of RNA, and sometimes they kind of mimic the way that they look in structure to other genes in evolution, maybe a duplication event. And so maybe if we're like looking at a blip on evolutionary time, maybe we didn't really mean for that to happen. And if we looked further in evolutionary time, that might be lost in the genome because we didn't really need it. Maybe it's just residual from some genomic rearrangements. But there's also a lot of evidence that some of these lncRNAs have crucial functions. So we'll see some examples of those later, some of which you've seen before. And we can understand some of the diverse functions that lncRNAs fill. So here are some things that you may be familiar with. So one would be the maintenance of linear chromosome ends. So you may also know this as the end replication problem. So this would be, of course, the problem caused by that lagging strand where we remove that RNA primer, and it shortens our telomeres in each round of replication. So this would be remedied by, of course, the enzyme telomerase, which we'll see on the next slide, kind of a deeper example of this. Telomerase has an RNA component, and this can kind of be thought of as a long non-coding RNA, even though it's part of that protein complex. And kind of just to add on to that, you can see the critical length of these shortening through subsequent cell divisions, ultimately reaching what we call a Hayflick limit, which will trigger senescence. This is kind of like the threshold of as short as it can be. Okay, so telomerase, we'll look a little bit more about that on the next slide. The other example you may be familiar with is X chromosome inactivations. And this is specific to mammals, and this is going to be individuals that have 2 X chromosomes. So hopefully you remember back to when we talked about sort of the chromosomal structure, we actually have to silence one of those. So we have an active X chromosome and an inactive one. And inactive one, this is kind of a fluorescent staining technique used for these different pieces of the genome, so different chromosomes. This one's going to be kind of inactive. It's not going to have, it's going to be like tightened down. We're not going to have transcription from it. So basically we just get transcription from one of the X's. This is the mechanism by which we get primarily female calico cats. So we'll see some calico cats a little bit later. This is next, similar thing. You can kind of see how it's tightened down here. This is DAPI stain, which stains DNA within the nucleus. It's a pretty common stain. If you work in a lab, you'll probably see this at some point. But we're going to see that this X chromosome inactivation actually mediated by long non-coding RNA. So let's revisit telomerase. So a lot of these discoveries, I'm not going to do this the level of justice that it should be done. But a lot of these discoveries we started out in Tetrahymena, okay, which is our model organism here. And in our Tetrahymena, we can see that these repetitive sequences are added to the chromosomes ends by an enzyme complex called telomerase. And just a little bit closer look at that, you can see we have, this is going to be the protein part of that complex, and we're going to have an RNA that's specific for this. Okay, that's going to be our telomerase RNA, but this is also we can kind of, in a way, think of this as a long non-coding RNA. Okay, we can also see how this helps protect our cells through various rounds of cell division, particularly in multicellular individuals through modifying the ends of cells that will become gametes. But we can really see the structure of this RNA. You can see sort of the tertiary structure. Here's the template part that'll be used, and then so here, and then some structural components as well. So this is going to, of course, be longer than 200 nucleotides, and it's going to perform a specific function. Okay, so we can really relate this back to our telomerase. So this RNA, we call this telomerase RNA TER, can serve as a template for nucleotide addition. And we're also going to use it as a scaffold to recruit the multiple proteins needed for the actual telomerase ribonucleotide protein complex. So let's revisit that idea of calico cats. Okay, there's a lot of jargon on this slide, but that's okay. We can see exist in action. So again, thinking back to that X chromosome inactivation, where we have one active chromosome and one inactive chromosome, we actually have a center embedded in this chromosome that's going to help us kind of mediate this process. In that inactivation center, which if you want to, you can look a little bit more closely, we're going to pretty much be dealing with the exist gene's RNA. See that we transcribe this exist RNA. Okay, and this is going to be very early during embryogenesis, so we have to silence one of these X chromosomes pretty early on. Okay, this exist link RNA can be 17,000 nucleotides long, which is a pretty long RNA. Okay, it's going to contain binding sites for a protein complex called the PRC2 complex. Just hang out, and we'll talk about that in a second. So that's actually going to help modify our chromatin. So we can see the transcription of our exist RNA. This is going to be our PRC2 complex, okay? And you can see the multiple binding sites. So it's going to help kind of coat this whole chromosome and recruit in that PRC2 complex. Okay, and this PRC2 complex is going to help us methylate histone 3 along this inactivated chromosome. So hopefully remember back to methylation. Methylation is, if you think just in and of itself, is pretty often associated with tightened down transcriptionally inactive DNA. So this exist RNA will actually keep pumping this out, coat the entire chromosome, and then bring in this PRC2 complex to methylate the whole thing, which you can see here kind of in pink. Okay, and so this is going to use the exist RNA as a guide to know which chromosome we're going to methylate and shut down. So this is a pretty cool way to do this. Okay, so again, always crediting people who discover these different long non-coding RNAs, and this was kind of in 1991. So this was, I think, in scientific time, this is a fairly long time ago that we actually knew about these. Okay, so this is basically, I mean, this answers a lot of questions about X chromosome inactivation, but also why cats are calico. Okay, so let's think about the way we might analyze these. So in our upcoming lab experiments, we're going to be kind of digging around through some sequencing data that was derived through that bridge amplification and sequencing by synthesis that we talked about in chapter 8. And instead of sequencing DNA, you can also use this type of technique to sequence all of the RNAs that come out of a particular tissue, organism, cell type. So we're going to be looking at the full transcriptome, all the transcripts produced at once. Okay, so that'll be something we're working towards in lab, but let's connect it back to some of these examples. So our next generation sequencing is driving the identification in functional analysis of these lncRNAs because we're finding out that they exist. And then we'll use a technique called, well, under the umbrella of reverse genetics. We'll be talking about reverse and forward genetics in later chapters. But our reverse genetics is basically kind of trying to think about, you have this RNA and trying to figure out how it relates to the phenotype. So our TER and our exist RNA were considered exceptions to the central dogma, the gene equals protein kind of hypothesis. You know, that one that goes all the way back to Beadle and Tatum. We're finding these RNAs that aren't coding for anything, so they're kind of outside that central dogma of a gene makes an RNA, makes a protein. So they're kind of considered exceptions in the sense that there's probably not a great deal of this going on. Okay, some people don't even consider our telomerase RNA to be a long non-coding RNA, even though it, by definition, is long and non-coding. We kind of think about it more like a maintenance or like ribosomal RNA. Okay, we found other long non-coding RNAs that are regulatory. Of course, I can always talk more about these if you want, but not included in this presentation. We've got things like hot air, mammals, cool air, cold air, and plants that are going to be really heavily involved with regulation. And we're going to see that they are involved with like PRC2 binding lncRNAs. So this high throughput sequencing is really allowing us to find out a lot of things that are going on. And so with the identification of more of these long non-coding RNAs, we're now able to start going back and saying, hey, do these have a function? And if so, what are they doing and how? So let's talk about 4 main functions that long non-coding RNAs can have. So one's going to be signal. So they can act as signaling molecules. So acting as transcriptional regulators in response to changes in the environment, okay, or in, you know, just whatever's going on in the cell, and often can be involved with regulating transcription directly. Another cool function of these guys is a decoy. So they can actually pull proteins away from other genes. So if there's going to be a protein that should be involved somehow with transcriptional activation, we can actually pull that away from the promoter. And there are other ways this can happen, but use this to sort of absorb molecules that would otherwise be signaling. So kind of like, almost like an inhibitor, if you want to think about it. It's not an enzyme, but kind of in that way, so acting like a sponge to absorb regulatory molecules. This can also target even those microRNAs we just talked about and sort of absorb those so that they can't get access to their target. They can act as a guide. So this can actually recruit proteins to particular chromosomal regions. Okay, so, and then we can get kind of, you know, remodeling happening here. By a guide, we're not talking about individual mRNAs, we're talking about chromosomes, like to places on the chromosome. So I would think of this as maybe more in the family of that exist RNA. Or they can act as a scaffold. So these scaffolds, again, help us facilitate both interactions between proteins that we need in a complex. We can bring in different types of proteins on the same purple lncRNA here, and we can also include binding to chromosomes or not, just depending on what we're doing here. So in this particular case, we're again looking at chromatin modification, but we can target other things too with the scaffolding mechanism. So these are 4 general ways that these work, and you can think of probably a lot of variations on this theme if you wanted to. So what we're going to be doing in lab for the next few weeks is learning to identify these specific types of RNA, the long non-coding RNAs, in the whole sea of transcriptome data. So we're going to be looking through experiments that were performed, this high throughput sequencing of RNA, and trying to find which would be long non-coding RNAs. So that will be kind of a project that we're working on for the next couple of weeks. We'll be using some online software and applications to do this. So it's important for us to at least have some idea of what we're looking for and some idea of the way in which these algorithms might go about identifying these in the whole sea of all of the other RNAs that could be present in an organism. So let's go ahead and actually look at a video of how this works. So let's be honest, this is a video from Bio-Rad, so they're going to try to like sell you a little something at the end, but I think this is a good representation of the activities that we're going to be kind of undertaking.
Long non-coding RNAs, or lncRNAs, are a newly discovered class of RNA transcripts. For the next couple of minutes, let's explore this intriguing field. Not too long ago, scientists thought of RNAs as only a simple, short-lived copy of our DNA, serving as a template for the synthesis of proteins. But their interpretation of the transcriptome was not without inconsistencies. Of the 3 billion bases of the human genome, only approximately 2% codes for proteins. Yet a much larger fraction of our genome is actively transcribed into RNA. Is this all just noise created by an overactive polymerase producing nonspecific transcription? Our understanding of the genome has advanced tremendously with improved genomic technologies that have enabled scientists to finally bring the picture into focus. Expression of non-protein coding transcripts across our genome is not noise, but rather the expression of tens of thousands of functional transcripts. But what defines long non-coding RNAs? lncRNAs are classified as transcripts over 200 nucleotides in length and share many traits with their protein-coding mRNA cousins, such as exhibiting intron-exon splicing, regulated transcriptional control, and being expressed in a tissue-specific context. lncRNAs, however, exhibit some unique attributes as well. Because the RNA molecule is functional and its structure will often dictate function, the secondary and tertiary structures of lncRNAs help determine their role. lncRNAs have been shown to act like a sponge or decoy for other RNA molecules. Alter mRNA splicing through interaction with splicing factors. Modulate epigenetic state by recruiting chromatin remodeling proteins. Recruit or block transcription factors to affect gene expression. While lncRNAs have so much to reveal to us about their functions, they do differ from mRNAs. lncRNAs often have little homology across species, so you must choose a model system carefully. They are typically expressed at much lower levels than mRNAs, so sensitive detection methods are required to measure changes in expression. They can exist in complex genome sites, between protein-coding genes, within genes, or even overlapping other genes, so specificity of the assay used for detection is crucial. Despite being a relatively new field, lncRNAs are gaining huge amounts of interest from researchers because they can play significant roles in our biology, such as cell development and differentiation, epigenetic regulation, and pathogenic disease processes, where they are being identified as new biomarkers or druggable targets for cancer, heart disease, inflammation, diabetes, and neurodegeneration, just to name a few. More discoveries are yet to come in this fascinating new field of research. Bio-Rad offers a complete lncRNA gene expression workflow with sensitive and simple-to-use tools for genetic analysis, including reagents, consumables, instruments, and analysis software. Come find out more at biorad.com and begin your lncRNA exploration.